Hello, my name is Natalia Marmo. I'm part of the CUNY Research Scholars Programs, and I'll be talking about antioxidants and polyphenols in commercially roasted and raw coffee. I'll go through a quick introduction, tell you about antioxidants and polyphenols, the objective of my research, and the steps that I took to obtain my results, as well as the overall conclusion. Antioxidants are molecules that fight off free radicals, and free radicals are molecules that harm our cells. Polyphenols are found in plants, and they can act as an antioxidant, neutralizing free radicals or those harmful molecules. Antioxidants and polyphenols are both known for their health benefits, and my research is focused on polyphenols in both commercial and raw coffee. Coffee is one of the main sources of antioxidants for Americans, so the overall goal of this research is to find the capacity of polyphenols in the different kinds of coffee using the Foley-Shakatao method. To obtain my results, I grinded and I sieved coffee beans to particle sizes that were less than 0.355 millimeters, and I used 0.5 grams of my samples to 10 milliliters of distilled water, and this was triplicated. Then the samples were in the mixer for 10 minutes and in the centrifuge for 10 minutes. I filtered each to gain filter that was then used to dilute at 5 different concentrations. And using the Foley-Shakatao method, I was able to obtain the absorbance, which was used to determine the polyphenol content. This graph shows the epic attachment standard curve. It's the relationship between absorbance and concentration. The epic attachment was used as a reference, and all of the polyphenols amounts was compared to the epic attachment extracted polyphenols. It is a pretty strong relationship. As you can see, the regression line is close to 1, and this equation above was the equation used to determine the polyphenol content by simply plugging in the absorbance over for the concentration and to gain the epic attachment equivalent per gram sample. Using the Grubbs test, the outliers were determined and about 1 to 2 outliers were removed from each of my data, and the error bars is the standard deviation. But this graph shows the raw coffee samples, the results for them. As you can see, Colombia showed lower content of polyphenols, which was about 50, compared to Ethiopian and Mexican, which were about 50 to 60, which essentially showed the higher content of polyphenols in raw coffee. Geographical impact could be possible since Colombian dish has showed the lowest polyphenol capacity. This image shows lab-treated coffee beans, which they were in a process to somewhat imitate roasting. As you can see, the longer they roasted, the darker the coffee bean got. This is the result of the lab-treated coffee. As you can see, the content of polyphenols were consistent as they stood about 60 and 70, but there is a significant drop at the 60-minute mark. You can see the polyphenol content dropped to about 50, and it shows that the longer the beans get, the polyphenol content drops. And it could be said that between 0 and 20 is light, 30 and 50 is medium roasted, and 50 to 60 is dark roasted. This image shows commercially roasted coffee between dark, medium, and dark compared to the lab-treated 60-minute mark. As you can see, the longer the beans go through a roasting process, the darker the bean color get. And this shows my results for that in comparison to the Colombian raw coffee, which is the coffee that showed the lowest polyphenol content. Light and medium commercially roasted coffee showed consistent polyphenol amounts, as it was about 40, compared to the dark, which showed the significant drop to about 30. And compared to the raw sample that showed the lowest content of polyphenols, it could be seen that raw coffee has the highest polyphenol content comparison to commercially roasted coffee. In conclusion, raw coffee showed higher polyphenol content, Ethiopian showing the highest, while Colombian showed the lowest. Raw coffee did have higher capacity, and roasting process may impact the available amounts of polyphenols in coffee. And when it came to commercially roasted coffee, dark showed the lowest capacity of polyphenols compared to light and medium. Thank you.